So, we've looked at the formation of the leading strand. Now we're going to look at the formation of the lagging strand. So, as it says, DNA polymerase is only able to add nucleotides to the free 3' end of an original DNA strand. Therefore, the DNA template strand has to be replicated in fragments, each starting at the 3' end of a primer. So, if we just go back a second and have a look at this diagram. Here's the leading strand, okay? And we know that the nucleotides can only be added to the 3' end of the original strand. So we know that in the leading strand, nucleotides would be added like this, in that order, and in that direction. And as this opens up, more bases are exposed, it continues in that direction. For the lagging strand, because the 3' end is at this end of the molecule, and these DNA bases are not exposed yet because they still have their hydrogen bonds intact, we have a bit of an issue. What has to happen then is that nucleotides can still only be added to the 3' end of the original molecule, but they can't get to the 3' end. So what happens is DNA polymerase will add them in fragments. Now what happens is you get, just go to the next slide, a primer to start the process is added. So you would have, say, a primer would be added here, and then nucleotides would be added on to the primer. And then as a little bit more of the molecule unzips, another primer would be added, and some more nucleotides would be added by DNA polymerase. Then as the molecule unzips again, another primer would be added, and some more nucleotides would be added again. And this keeps going until you have all these little fragments, which are nearly making up a strand, but they have to be joined together. And join the fragments together, an enzyme called ligase is used, and ligase is almost like a glue that basically joins the fragments. So what we're going to do is I'm going to show you this, follow this link, takes us to BBC Bite Size, and they have a good interactive diagram. Why is that? Here we go. So have a look here. This is the diagram down at the bottom of the page. This is our DNA molecule. First of all, hydrogen bonds break and it's unwound as well. Leading strand forms, as we've already discussed. The important one we're looking at now is the lagging strand. And you can see here that the green parts represent a primer, the blue parts represent the nucleotides, and you can see that a primer has formed and nucleotides have joined on. Primer is formed and then nucleotides join on, and the same again. And that would keep going as this DNA molecule is unwound. What happens then is these primers are then replaced by nucleotides, and they are joined up using the enzyme ligase until you get a full new strand, and this is called the lagging strand.